Next class in metabolism and metabolic regulation is lipoprotein metabolism. In this lipoprotein metabolism, we are going to study about the different lipoproteins. They classified as VLDL, LDL, and IDL and HDL. So all the four components we are going to study, and how the cholesterol is involved in the synthesis of HDL is also going to be elaborated, and how it is regulated, the cholesterol biosynthesis, how it is regulated, and what are the anti-metabolites of cholesterol biosynthesis, and especially the HMG-CoA reductase inhibitors. So everything we are going to cover in this lecture. We can go with the first slide. We already discussed about the cholesterol. It has several fates. Okay, that is synthesis in vertebrates takes place in the liver, and export of the cholesterol in biliary. Okay, that is the biliary cholesterol, bile acid, and cholesterol ester. So these are all the three export forms of the cholesterol. And membrane synthesis or a precursor for steroid hormone and vitamin D. Okay, that is also a major function of cholesterol. They are all carried in the blood plasma as a plasma lipoprotein. So we are going to study about this one now. And a different combination of lipids and proteins produce the particles of different densities, ranging from Kylo Macrons to high density lipoproteins. So lipoproteins are the combination of lipids and proteins. That is what is called as lipoproteins, and it will produce the particles of different densities. So the size may vary, the density may vary. So we can see all the four different types. Okay, that is the first one is Kylo Macron, the biggest one. The density is very high. Okay, that is 1.006. Okay, that is the density, but the size is maybe the biggest one with okay, that the lipoprotein. Okay, this is the biggest one. I'll show the comparison of the sizes also. And protein only 2%, and phospholipid 9%, free cholesterol 1%, cholesterol esters 3%, and triacylglycerol 85%. The majority is the chylomicron. Only the dietary triacylglycerols are maybe the packed in the form of chylomicrons. The origination of this one is in the small intestine, and the VLDL is 0.95 to 1.006. You can see the majority is again triacylglycerol, but this is reduced to 85 to 50. You can see the phospholipids and proteins are increased here. Next one is LDL. LDL is formed from VLDL, and this LDL, the VLDL is synthesized by liver, and LDL is from VLDL. Okay, that is the triacylglycerol levels are decreased so much, and you can see the proteins and phospholipids are increased much when compared to the VLDL. And next one is HDL. The concentration of protein is very high. At the same time, the cholesterol esters and the free cholesterol is also very high, and the very less quantity of triacylglycerol may be there. So this is the indication of HDL, because HDL is very important to reduce the concentration of cellular cholesterols and cholesterol esters. So that is a very important one. So the deficiency of cholesterol HDL may lead to many problems like the heart problems, and also deposition of fat on various areas of the arteries. So this may lead to a lot of complications. So this is the general structure of the lipoprotein. You can see here the lipids and protein combination, and the protein is here as the ApoB100 as an example of the protein given here, and you can see the free or unesterified cholesterol, and the cholesterol esters are embedded here within the core component, and the triacylglycerols are here. Okay, this E is nothing but triacylglycerols. Next, coming to the size, you can see here this is the scanning electron microscope of the magnification capacity is 60,000 maybe times of magnification, and you can see here the size of the chylomicron. Okay, at the same time you can see the VLDL is 180,000 the magnification. You can see the size. So this is smaller than this one. At the same magnification capacity, that is 1,80,000 magnification, X magnification, you can see LDL is this much size, and HDL is very small. and you can see the size of chylomicron, and this chylomicron is a very big structures, and maybe the magnification is not 1,80,000. This is including in 60,000 magnification we can see the chylomicrons. The largest of the lipoprotein, less dense, high content of triacylglycerol and dietary fatty acid to target the tissue, that is ApoC2 protein. The Vldl, next one is the transport triacylglycerol from liver to muscle and adipose tissue. The composition of the protein is apolipoprotein C2. The Ldl, very rich in cholesterol and cholesterol ester. Receptor mediated uptake is going to function as a transport mechanism, and HDL originate in the liver and small intestine as small and very small particle, and the protein rich particle. LCAT on the surface of the nascent HDL particle converts cholesterol and VLDL remnants to cholesterol ester, begin to form a core. So this mature HDL return to the liver where cholesterol is unloaded. So we can see the cycle of the pathway leading to the synthesis of cholesterol esters, and how it is incorporated in all the lipoproteins, and how chylomicron is going to be formed, and what is the each and every the pathway involved in chylomicron, VLDL, LDL and HDL also separately in detailed manner. But before going to that, we can have an overview of all. So this pathway. So this is overview. This is not a final one. Just to have a look. And this is the one. The first one is the chylomicron synthesis from the intestine. The dietary lipids. For example, a person eat a particular substance. There's a diet, and this is going to the small intestine, and the small intestine we should absorb the fat and forms chylomicron. The chylomicrons may be synthesized inside of the small intestine. So the dietary lipids are packaged into chylomicrons. Much of their triacylglycerol content is released by lipoprotein lipase to adipose tissue and muscle tissues during the transport through the capillaries. If it goes through the capillaries, for example, if it is absorbed by the blood circulation, and this is within the blood circulation, that is the capillary of the blood circulation, just you can see an enzyme, that is a lipoprotein lipase, an enzyme involved in that, that will liberate free fatty acids from the chylomicrons. Chylomicrons having triacylglycerol. From the triacylglycerol, the glycerol particle is removed, and the free fatty acids are liberated, and that is absorbed by mammary gland, muscle and adipose tissue. So all the three glands which need more amount of energy, at the same time there's more amount of fatty acids also. And after the liberation of free fatty acids, this is converted into VLDL, and this VLDL is maybe the chylomicron remnants and VLDL. This will go directly to the liver, and again this liver maybe that will coat additional apolipoproteins and liberates VLDL. So again this VLDL come to the capillary tube and again releases the free fatty acids by the action of lipoprotein lipase and forms this LDL. So this is ideal. Ideal means intermediate density lipoprotein, and in between this will be forming LDL. So this LDL is also going to bind with the liver, and so this will form again maybe metabolized further in the liver, and again this HDL precursors from the liver and intestine that will be in the extrahepatic tissues. That means extrahepatic tissues means and maybe in the small intestine. We can say an example. There the HDL is formed. So this HDL again go and bind to the liver. So ultimately the HDL precursors from the liver and intestine will synthesize maybe given to extrahepatic tissues to synthesize HDL. The HDL will reverse the cholesterol transport. That means it will reduce the cholesterol concentration in the blood circulation. So the chylomicron remnants containing largely a protein cholesterol are taken up by the liver, and endogenous lipids and cholesterol from the liver are delivered to the adipose tissue. Whatever I told that is a given here, and maybe this is the overall view of the lipoproteins. First we can start from chylomicron from the right side. So this is the chylomicron. You can see the size, so that matters, and the area of synthesis is intestine, the small intestine, that generate chylomicron. This is composed of highest concentration of triacylglycerol and lowest concentration of cholesterol also. and this functions to deliver dietary, that is exogenous triacylglycerol to the peripheral tissues. The main function is to deliver or transport fat, that means the free fatty acids to the tissues like the mammary gland, liver and adipose tissue also. Next one is VLDL. Liver is the part involved in generation of VLDL, and this VLDL is composed of high amount of so this is the highest and this is the high amount of triacylglycerol and low cholesterol. This is the lowest cholesterol and this is low cholesterol, and this is functions as deliver the endogenous triacylglycerol to peripheral tissues, the same function. And next one is from the VLDL, LDL will be formed. So that means the liberation of more amount of the free fatty acids by lipoprotein lipase of small intestine, that will lead to synthesize the LDL. So the LDL is composed of low triacylglycerol. Just you can see highest, high, and this low triacylglycerol. Next one is in HDL, the lowest one. Okay. So this low triacylglycerol and highest concentration of cholesterol also there. The functions to deliver cholesterol to the peripheral tissues and to the liver. So next one is HDL. So that is liver and intestine that will generate HDL, and is composed of lowest amount of triacylglycerol and high amount of cholesterol. So this is functions to deliver the cholesterol from peripheral tissues to the liver for elimination. So that will collect all the cholesterol and maybe give it to the liver for the further elimination. Elimination means either by bile acid, that may liberate into the intestine again. Okay, so this is the overall map of lipoprotein. What is the size and where it is originating, and how it is how it is functioning, and what is the composition of each and every lipoprotein. All the four is discussed here. So this may give the clear output about the overall view of the lipoproteins to you. So next we can have the metabolism of chylomicrons. So this is explained in this chart. And this is a very colorful chart from the book Lipincot's Illustrated Biochemistry, and that is a wonderful book with a lot of colorful illustrations. And you can see here the small intestine. This is small intestine, and this is nascent chylomicron. Nascent means within the cell it is called as nascent. So you can see here the triacylglycerol is very high when compared to the cholesterol esters and cholesterol, and the coated with the protein, that is ApoB48. We already shown the protein, that is, just you can have a look. This is the type of protein. That protein is present as the integral protein, and this integral protein means that is maybe penetrated through the membrane and maybe present on the surface also and inside also. That is called as integral protein, and this protein is ApoB48. That is a protein. And to the nascent chylomicron, ApoC2 and ApoE. So further two proteins. This is the first protein, and ApoC2 and ApoE are transferred from HDL to nascent chylomicron. So from where the C2 and ApoE are coming means from the HDL. So HDL is the donor for C2 and E. So this C2 and E are coated with nascent chylomicron. Now this is forming a full-fledged chylomicron. Now the chylomicron is the combination of three apolipoproteins. The protein, this apolipoprotein B48, apolipoprotein C2, and apolipoprotein E. All the three are there. Don't forget. All the three are very important, and you should remember the names of apolipoproteins. And by the blood circulation, it goes to the capillaries, and capillaries having the free lipoprotein lipase enzyme. So that means extracellular lipoprotein lipase activated by ApoC2 degrades triacylglycerides in chylomicron. So this is chylomicron. So if it is goes to the capillaries and by the action of lipoprotein lipase enzyme, so that is an enzyme that liberate free fatty acids and glycerol, and this glycerol is utilized by the liver for further synthesis of triglycerides. And this free fatty acids utilized by the tissues, example adipose tissue or mammary gland or whatever. And this is very, very important. And so this synthesis of free fatty acids are maybe the major function of the chylomicron, and this free fatty acids are liberated in the tissues, and especially the adipose tissue, mammary gland. I already told this one. And The next step is maybe after the liberation of free fatty acids. So this one will be joining with Apo C2 is returned to the HDL. That is, Apo C2 is there because Apo E involved in the binding process, and maybe further Apo C2 is there. Apo C2 is again returned to the HDL, and remaining thing is Apo E and Apo B48. So chylomicron remnant, this is called as chylomicron remnant. You can see the size, and this size is maybe minimized to this one. This is called as chylomicron remnant. Don't confuse with the nascent chylomicron, that is different. Nascent means the pre-form of chylomicron, that is called as nascent chylomicron, and this is chylomicron remnant. That is remnant means the product after the liberation of free fatty acids. So the chylomicron remnants having apolipoprotein B48 and apolipoprotein E, and maybe the chylomicron rich, that is cholesterol ester rich chylomicron remnants bind through the apolipoprotein E to the specific receptors on the liver where they are endocytosed. That means apolipoprotein again, there is a receptor, and this receptor will bind to the liver cells during the circulation, and if it is binding to the receptor automatically, that is endocytosis. That means endocytosis means that there will be engulfed completely to the liver for further production of VLDL. So this is the life cycle of chylomicron. CM represents chylomicron, and TAG represents triacylglycerol, and C means cholesterol, and C means cholesterol esters, and ApoB8, ApoC2, and ApoE or the apolipoproteins found as a specific protein components of plasmolipoproteins. The size it is not comparable, but not scalable. Don't compare this one. This is very huge, and when compared to the chylomicron, so don't compare the size and all. This is the pictorial representation or illustration of the pathway. Next we can go to the life cycle of that is the metabolism of VLDL and LDL. And so here you can see here, this is a nascent VLDL. Nascent VLDL, that is a pre-form of VLDL within the liver. And now this ApoC2 and ApoE from HDL is going to be bind again. So you can see here this nascent VLDL already having ApoB100. So don't confuse with chylomicron. Chylomicron is having ApoB8 already in the nascent, and from HDL C2 and E are going to be attached. So 3 apolipoproteins will be there. And the next one is here in VLDL, already available one in the nascent VLDL, that is B100. To that C2 and E will be given by HDL. So C2 and E will be attached, and now this is the 3 combination of apolipoproteins, that is apolipoprotein B100 and apolipoprotein E and apolipoprotein C2. And the name is VLDL, so very low density lipoprotein. And this ApoC2 and ApoE are transferred to the HDL to nascent VLDL, and the nascent VLDL is converted to VLDL. Now the VLDL is going through the capillaries, and these capillaries again, the previously I told, lipoprotein lipase take away the free fatty acids, and glycerol transferred to the liver, and remaining remnant is called as IDL. IDL means intermediate density lipoprotein, and further apolipoprotein C2 and ApoE goes to the liver, that is HDL, and removal of these two, that is C2 and ApoE, remaining is apolipoprotein B100, that is called as LDL. So this LDL having the receptor, that means this LDL binds to specific receptors in extrahepatic tissues on the liver where they are endocyted, either in extrahepatic tissue and also in the liver. That means either muscle or liver. They have in the receptor, and this receptor is very specific for apolipoprotein B100. It specifically binds to the receptor of the muscle and also the liver for further maybe endocytosis and then also further metabolized. So this is the fate of synthesis and degradation of VLDL. And next, coming to the LDL synthesis, LDL uptake. So this LDL is taken back. That means you can see here, this is LDL. So this pathway, how it is going to be endocytosed in liver cells and extrahepatic tissues. Maybe this is the explanation for that. So we have 2 components, LDL and ACAT, acyl CoA cholesterol acyltransferase. These 2 are very important. LDL is this is LDL already having the B100. And inside this is cholesterol ester is there, and this lipoprotein B100 having the receptor on the surface of the liver and muscle, You can see here this is clathrine coated pit, and this clathrine coated pit is ready for taking endocytosis, and without clathrine, okay, that will not form the pit like something, and this will form a pit like in system, okay, by the mediation of the clathrine. And this clathrine along with the receptor is already bound receptor, okay, the receptor, okay, of this apolipoprotein B100 is already there, and this receptor is ready, okay. This is simply called as Ldl receptor, and due to this is going to be attached, and immediately after the attachment, okay, this endocytosis may be formed, and now the clathrine coated vesicles are formed. You can see the white dots are clathrine coated vesicles, and this vesicle may be fusing with the lysosome, and this lysosome may liberate this one, that is again further Ldl receptors are not completely digested, and that is again recycled to the cell surface, and remaining, okay, this one, the lysosome fused one, okay, that is fused with the Ldl, that liberate fatty acids, amino acids, and also the cholesterol. So this free cholesterol, the cell goes to the cell membrane, or used for the steroid hormone synthesis and bile acids. And by the action of ACAT, just now I told ACAT means acyl-CoA cholesterol acyl transferase. Acyl transferase means the cholesterol to cholesterol ester formation, and the cholesterol esters are not toxic form, that is not lethal, and that may be stored further. The storage of cholesterol esters, and you can see here this the oversupply of cholesterol. If the cholesterol concentration is very high, okay, that is there, automatically that will inhibit two places. That is, one is synthesis of Ldl receptors, that is blocked, and second one is the synthesis of cholesterol is also blocked by HMG-CoA reductase enzyme. The product the cholesterol itself, that will inhibit. That is why it is called as feedback inhibition of the substrate, and also, okay, this one will go and bind to the DNA, and I'll explain to you, okay, how it is going to be stopped, okay, later. And the mRNA synthesis for Ldl receptor is stopped, and if it is stopped automatically, okay, there is no mRNA is available. If there is no mRNA, there is no protein synthesis, and if there is no protein synthesis, there is no receptor. Once if there is no receptor, and the receptor will not be available for there, okay. So that is why, okay, this Ldl receptor synthesis also involved with the concentration of cholesterol, okay, that may play a role. If the cholesterol concentration less, it need a cholesterol. That is why it will facilitate the DNA to synthesize mRNA, okay, for the Ldl receptor formation, and all the Ldl receptors are packed completely and is maybe transported to the Golgi apparatus, and Golgi apparatus are vesicles, and vesicles too fused with the membrane, and membrane it is coated as a Ldl receptor. Now it is ready for taking Ldl. So this is the pathway of Ldl. Next, coming to the HDL. So this is a little complicated when compared to other, because it's entirely different one. No lipoprotein lipase is involved here. Hepatic lipase is involved, and other two enzymes also there. So we can see the abbreviation PC means phosphatidylcholine. So we already studied it in the phospholipids, and lyso-PC means lysophosphatidylcholine, and LCAT lecithin cholesterol transferase, and CETP means cholesterol ester transfer protein, and ABCA1 means it is a transport protein for free cholesterol. So the size is not comparable, okay. Here they gave a note for convenience. The size of the VLDLs are shown smaller than HDL, whereas VLDLs are larger than HDL, okay. VLDLs are larger than HDL, and HDLs are very small, very small when compared to VLDL. Okay, I'll show the picture because then only you can understand. You can see here this VLDL, the size, and the HDL size. So don't compare the size given here, okay, that is the representation. So that is the pictorial representation or illustration to understand the concept. The first of all, okay, this HDL is synthesized by the liver and the small intestine, and this is okay, discoidal, okay, nascent HDL, okay. This it is not a final form of HDL, and because this HDL, okay, having ApoA1, so this ApoA1 is a part of discoidal nascent HDL, and due to that, okay, maybe with the concentration of the cholesterol, okay, within the HDL, and due to the action of ABCA1, the ABCA1 is a transport protein. So this transport protein, okay, that will liberate, okay, lot of free cholesterol. So this is C means free cholesterol, and transported to the HDL. So that is with the concentration of cholesterol taken up by HDL, and at the same time, so this process is facilitated by the PC to lyso-PC. So this is a reaction, okay, catalyzed by the enzyme LCAT. LCAT means lecithin cholesterol transferase. So lecithin cholesterol transferase, okay, this ApoA is inducing, okay, this LCAT, apolipoprotein A is inducing the LCAT, and this LCAT is the enzyme, okay, that will convert the cholesterol to cholesterol ester, and cholesterol ester is packed inside the HDL. And the PC is converted to a lyso-PC also, okay, by this reaction. We can see, okay, what is PC and what is lyso-PC. PC is phosphatidylcholine and lysophosphatidylcholine. Okay, this is phosphatidylcholine is converted to lysophosphatidylcholine. Now we already studied about what is phosphatidylcholine and lysophosphatidylcholine in phospholipid metabolism. And due to this, the cholesterol is attached with cholesterol ester molecules are attached, and cholesterol ester is deposited within this HDL. Okay, this is called as mature HDL, and this HDL again receives as much of the cholesterol, okay, by the free cholesterol, okay, from the cells, the peripheral tissues, and again, okay, this is by LCAT only, because without LCAT there is no transfer of cholesterol into the HDL. So the LCAT deficiency may cause elevated levels of free cholesterol in the blood circulation that may be exceeding, okay, more than 200 to 210, and this may create a lot of complications in the metabolism, that is especially in the heart related issues or circulatory disorders. So this enzyme is very, very important, LCAT, that is a lysophosphatidylcholine. So this LCAT means, okay, this LCAT, lecithin cholesterol transferase, that is LCAT, lecithin cholesterol acyl transferase enzyme. This enzyme is also involved in the conversion of cholesterol to cholesterol ester and transport also. Okay, now this HDL3 is formed and HDL2 is also formed. You can see the size of the cholesterol ester is increased. That means the transport, the size is the cholesterol ester size is increasing, okay, due to the uptake of cholesterol esters from cholesterol, and the size is increased now. More amount of cholesterols are taken up. Okay, now the HDL2 reacting with CETP. CETP means is nothing but, okay, the cholesterol ester transfer protein. Cholesterol ester, that means the cholesterol esters are transported, okay, to we can see here the composition of cholesterol esters are very less in VLDL, and this VLDL will react with HDL and take away all the cholesterol esters to VLDL form. And now this is going to the binding of the domain, okay, that is SRB1 domain, and this is again maybe endocytosed, okay, within the liver, and cholesterol and cholesterol ester will be metabolized, okay, by the liver. So this is the overall pathway of HDL, and HDL is very important when compared to all the four among the four, and HDL involved in the conversion of cholesterol to cholesterol ester. Next, coming to the cholesterol biosynthesis is regulated at several levels. Okay, the very famous one is HMG-CoA reductase. Okay, this reductase enzyme is inhibited. This already we discussed about that one, and that is the rate limiting step, okay, in the metabolism. That is only enzyme controllable. So coming to the nucleic acid level, or otherwise the DNA level regulation. So sterol regulatory element binding proteins are there. Okay, sterol regulatory element binding protein. You can see here, this is cytosol and this is endoplasmic reticulum. Within the endoplasmic reticulum, so there is a protein is present, that is SREBP. SREBP is that's called as sterol regulatory element binding protein. If the concentration of the cholesterol is maybe if the concentration is normal, automatically you can see here the migration to the Golgi apparatus complex. Okay, this will be going and binding with in the form of Golgi apparatus, and the sterol binds with the ACAP and prevents release of SREBP. So this SREBP and this is not going to be liberated, and because this is liberated means this segment is very, very important segment. This will go and bind to the DNA. If it is bind to the DNA, okay, inside the nucleus, means automatically the transcription of target genes are activated. That means maybe if SREBP cleaved by the protease and enter nucleus, leading to the activation of target genes. Okay, this is the regulation, and so this regulation occurs by the cleavage of first protease, and this protease is cleaved, and this one is liberated now, and this one is liberated again by the second cleavage of protease. This is the first cleavage and second cleavage, okay, here in this part, Okay, and this is from the C terminal to this terminal. Okay, this part is going to be cleaved, and this one is liberated now. The release of the domain of SREBP migrates to the nucleus. Because it is small protein that may easily cross the nuclear pore and nuclear membrane pore and automatically go and bind to the DNA, and the transcription of target genes are activated. The transcription means the conversion of DNA to mRNA that is going to be activated. So this is the molecular, that is nuclear level regulation of cholesterol biosynthesis. And this is the hormonal regulation. This already discussed. We already elaborately discussed about this one. That is, insulin induces this enzyme HMG-CoA reductase. Glucagon that is suppresses, and this is feedback inhibition. So that is the multi-step pathway. The stimulates proteolysis of HMG-CoA reductase enzyme. Okay, this one is inhibits the synthesis of mevalonate, and the cholesterol itself, cholesterol esters are formed from cholesterol, and this is induced by the cholesterol. Okay, this is the complete regulation of cholesterol and cholesterol ester formation, either by enzymatic, nuclear, and the feedback inhibition. So these are all the substances used as a molecular mimicking substances, used as a inhibitors of HMG-CoA reductase, and this is the enzyme HMG-CoA reductase. This is the enzyme. So this enzyme, the substrate is the mevalonic acid. This is mevalonate, and you can see the structures. These all the structures are very similar. That means compactin, simvastatin, and provastatin and lovastatin. The statins are very famous one. That will inhibit, that is mimicking the structures of this one, but it is not a substrate. This is attached. This one, if it is mevalonate, that may be forming cholesterol, but without mevalonate, only the similar structures are there. Means this will go and bind to the active site as a competitive inhibitor and inhibits the enzyme. If the enzyme is inhibited, there is no synthesis of cholesterol. And further, the fate of cholesterol, just you can see all the maybe the hormones, that is steroid hormones are synthesized. The cholesterol is synthesized to maybe used for the synthesis of pregnenolone and progesterone to testosterone, cholesterol, the cortisol is a glucocorticoid, and testosterone to estradiol. These are the testosterone is a male sex hormone, and progesterone is a female sex hormone, and corticosteroid, mineralocorticoids are the corticosteroids involved in the complete regulation of cellular metabolism, and aldosterone that is also regulate resorption of sodium, potassium, and also the chloride ions in the kidney. And the corticosteroid, that is the glucocorticoid, affects the protein and carbohydrate metabolism and suppresses immune response, inflammation, and allergic responses. Estradiol, that is maybe similar to testosterone, the male and female sex hormones, influence secondary sexual characteristics and regulate female reproductive cycle. So that's all about the fates of cholesterol, and this is further detailed one. The cholesterol, just you can see, this is the backbone. So delta 3 and isopentenyl pyrophosphate. So this is the fragment going to be polymerized to cholesterol. We already had very in-depth maybe the study of biosynthesis of cholesterol. So this is the one. This is used in the synthesis of rubber and photal chain of cholesterol, chlorophyll, and dolichol phosphates can be formed. Quinone of electron carriers and ubiquinone, plastoquinone will be formed from this substance, and isoprene molecules will be formed as the polymeric substances, and the plant hormones, abscisic acid and gibberellic acid will be formed in the plant, and carotenoids will be formed. Vitamin K and vitamin E all formed from isopentenyl pyrophosphate. So this is a very important one. From the cholesterol, vitamin D will be formed, bile acids and steroid hormones will be formed. So the cholesterol metabolism is very important, not only for the synthesis of cholesterol, and in between, this one, that is delta 3 isopentenyl pyrophosphate, is a precursor of so many very important metabolites, not only in animal cell and also plant cell. So with this, we can come to the conclusion of the lipoproteins, and lipoproteins are elaborately discussed in this lecture, and all the different types, and what is the fate of each and every lipoprotein, starting from Kylo Macron, Vldl, Ldl, and HDL, and how it is synthesized and how it is metabolized, and what is the regulation of cholesterol also, that is also discussed. Okay, thank you.